We can still wait a minute or so. Most are coming a little bit later, at least at our place.
Just putting in there cups of coffee and cups of tea.
Yeah, yeah, I already had a few, so it's okay.
Already by 8. Yeah.
So this is very much a cryo-EM membrane protein consortium, if I understood it correctly.
Correct. Yes. Yes.
Very good.
Yep. Yep. So that's
Consortium, which is to the point. That's good.
Yep. And yeah, so we're kind of like Australia wide in terms of the main nodes are Monash, University of Melbourne, Weehai, and University of Wollongong. But we do have members who are like in Queensland and ANU and all around University of Sydney. So it's quite a broad group. Yeah, but the
And is there connection with Adelaide also or not?
We don't have Adelaide members, no.
No connection. Okay, good, good.
So yeah, but yes.
It's amazing technology. We are talking to each other, right? And this is like from one corner to the other.
Yep, yep. That's the good thing about having only online seminars. We can get speakers from all around the world.
Yes, 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 because there are important distances between Europe, for example, and Australia. So it's not bad. This online idea is very good, actually.
Yep. We even had some from Europe at like 6 a.m. their time. So we're very grateful for that very early start for their days.
Okay.
But yeah, it makes it possible, which is good. All right, maybe, I don't know, just maybe one more minute, if that, or just.
Yeah, yeah.
Yep, I'm going to just.
So you have a large Greek community in Melbourne, apparently, right?
Very large Greek community in Melbourne. I think it's like the third after Athens or second after Athens. It's a very
Okay,
large
okay,
Greek community.
okay. Interesting.
Yes, you'll find plenty of galaktobudico and baklava and pastitso everywhere.
Yeah. So we wait still, or
I reckon we can go. So, Bronte, do you want to start off? Thank you.
Sure. So today we are very fortunate to hear from Professor Dimitrios Fotiadis, who is the Managing Director at the Institute of Biochemistry and Molecular Medicine and the Vice Dean for Research at the Medical Faculty of the University of Bern in Switzerland. So his research focuses on the structure and function of proteins with a particular focus on membrane proteins, including membrane transporters, GPCRs, light-driven ion pumps, and viral protein complexes. Using his extensive experience in cryo-EM, X-ray crystallography, and AFM, his work has been able to elucidate the structure, assembly, and functional mechanism of various membrane proteins in a detergent solubilized state and in the native environment, the lipid bilayer. So today we'll be hearing from Dimitrios on his work that has led to insights into how glucose is translocated across the membrane by a bacterial membrane transporter, as well as the structural basis for how light-driven protein pumping is achieved by a bacterial rhodopsin. So please join me in welcoming Dimitrios, who can now take it away. Thank you.
Thank you very much for the kind introduction. So as you said it, summarized it, I love membrane proteins, so that was my first love, right? And we are working with membrane proteins, but at very different levels. Just as a general information, today you will only get some, let's say, structural aspects decorated with a little bit of biochemistry and biophysics, but we are also applying them, for example, to build up new molecular systems. Just because we know them so well, we have them in hands in milligrams, so we can use them as building blocks. But that will not be the topic today, but just as a side information. So when I got this invitation and I asked Tracy about how long it will take, she told me about 40 minutes, 45 minutes. So I said, yeah, well, I think I have to take more than one topic, and I prefer to talk about a few topics, but a little bit more as an overview instead of going into one topic and very, very detailed at the molecular and the here and H bond and there. So I think it will be good for the audience. So I decided to talk today about transmembrane glucose transport in a first part, and about light-driven proton pumping in a second part, right? And yeah, I would just split it into two, and I would just straight start with the first part, namely about this transmembrane glucose transport. So just a very brief summary. We all know how important glucose is. It's very abundant monosaccharide in nature, very important building block. It is really the important immediate source of energy for many organisms. However, when you look at the structure, you see immediately that it's very hydrophilic. So you see all this hydroxyl groups, right? So it's very hydrophilic. It will not pass the lipid bilayer, the plasma membrane. That's why, just as two examples, in humans, this is the import into cells is facilitated by GLUT transporters, right? And or secondary active transporters, which are coupled to sodium, like the SGLT1. So today we will have more focus on transport in bacteria. And there, just to say it already, there is a very distinct special transport system, which is the so-called phosphoenol pyruvate dependent phosphotransferase system, abbreviated PTS. So I will use the abbreviation in the coming minutes. And this couples the glucose uptake with its phosphorylation. So now let's look at bacteria, right? Only, right? Here we have different systems for the uptake of solutes, inclusive glucose. So we can have a primary active transport, for example, through ABC transporters of solutes, or we have secondary active transport, for example, through major facilitator superfamily transporters, proton-driven, as you see here, not the case in human. And then we have solute sodium symport transporters, similar to also, as I said before, in humans. This is also secondary active transport. And then we have this special one, right, which is this group translocation transport, which uses phosphophenol pyruvate, takes the phosphate group from this molecule. The result is that you have pyruvate and also phosphorylated glucose. So let's have a look at this PTS system, right? In bacteria, I will specifically talk today about the PTS glucose transport from E. coli. So historically, it was named into two systems, like clusters of proteins. So we have system 1 and to system 1. The part of the system is the energy coupling enzyme 1, right? E1 abbreviated. And this histidine containing phosphocarrier protein. So what happens is that this E1 takes over the phosphate group of the phosphoenol pyruvate. You get pyruvate, and then it's phosphorylated. It has bound the phosphate group. This phosphate group is then transferred to this second protein I just mentioned, and this system 1 is also a little bit the so-called general part. So these are general components of this PTS system. Just to say, because this PTS system contains transporters which are also specific for other sugars. So many different transporters with different specificities. But of course, we were interested in glucose because of its high importance, right? But this part is used by all of them. So that's system 1, the general part, let's say. And then it becomes more specific, right? Then you have the so-called 2A protein, which takes over the phosphate from the histidine-containing phosphocarrier protein, right? And this 2A, and now we are in E. coli, in the glucose transport PTS system, right? This is also soluble in this specific case, right? And then this gives then over the phosphate to the 2B protein. And the 2B protein is part of the transporter. Okay, this is fused at the C-terminal part. It's part of the whole gene, right? So you have the 2C transporter, which is the membrane protein, which transports glucose. And immediately after, right, at the cytoplasmic site, you have this domain, this 2B domain or 2B protein. Because why both are correct? Because depending on the sugar system, this is part of the transporter or not. It can also be soluble. And there are also other transporters which have even this 2A fused to it in the same polypeptide chain. So just to see how heterogeneous this system is, right? But let's stick now to the glucose system. Okay, so now we have this 2B, right? And this is phosphorylated. And once the glucose comes in, right, the glucose is phosphorylated and like this trapped, right? Like in the other systems, you know, the hexokinase traps the glucose and it cannot go away, and the gradient is kept so that more glucose is coming into the cell. And this system part here we call system 2. Okay, so that was a little bit the general introduction. So we learned now that the transporter is very special because it has a domain, right, which binds a phosphate and transfers it during uptake to glucose, so that you have glucose 6-phosphate. Okay, a little bit of history. We started very early with this project. I think we started in 2009 or something like that, so several years ago. And I would just like to briefly show how much time and effort we invested here, right? And how painful this was. So we started this, we published in 2011, but as said, we started in 2009 or end of 2008. And we purified the protein, right? And we didn't purify the whole protein, we only purified the transporter. It was only the C domain, this part here, which is the membrane protein without this part. Okay, and when you look at it, we could purify it, was pure, and just doing negative stain electron microscopy, we could see that it is a dimer. You see this eyeglass-like structures, unsymmetrized, symmetrized. Here you see raw data, so you really can see it's a dimer. Okay, that was already something, or it's a dimer. And then we tried to make two-dimensional crystals of this protein, because I come from the lab of Andreas Engel in Basel at the Biocenter at that time, and that's where I started structure biology, single particle, lower resolution, and 2D crystals for atomic force microscopy and electron crystallography. So we managed to get crystals, and we managed to get the topography. With atomic force microscope, as you see here, you get only the topography. You don't see inside the protein. So you don't see inside the protein. That I will switch off my email, otherwise we will get a lot of strange noises during the talk. So we could get two-dimensional crystals. This is a topology, and you see dark means the lipid bilayer surface, and what protrudes out is getting bright. So we really see the topography of the protein. It's a two-dimensional crystal, and we see, yeah, well, it contains two protrusion. Again, it's a dimer, and here we see we have a kind of two indentations looking very similar also to the negative stain. So that was already a highlight at that time, because not much was known. And then, of course, you wanted to get resolution, right? And try to grow three-dimensional crystals. That's the best we could get, right? Also, again, without the B domain, only the membrane part, because we said, yeah, well, the B domain will be flexible, so it might only make problems trying to grow three-dimensional crystals. So that's the best we could get. Data set to about 4 angstroms. You see here the outer shell. Well, yeah, not very promising, no faces. So That's it, right? It was not good. So we continued a little bit with two-dimensional crystals, which were getting better and better. Here we could get some projection maps. You see, you only say a projection into the two-dimensional crystal, which is embedded in a lipid bilayer. So we were very happy to see it already at 4.5 angstroms. You see in projection the dimer, right? You see that we saw kind of a dimerization domain here, and here some domains which are supposed to bind the substrate. So that was the state. We could get projection maps in the presence of D-glucose and L-glucose. D-glucose is bound, L-glucose is not bound, as you will see later. But that was just to embed the protein to do the electron crystallography. So that was the state, right? Not very satisfactory. And then cryo-EM came. Well, cryo-EM we were doing also at that time, but the nice cameras came and the better microscopes, but in particular the cameras made a difference. So we said, okay, we have everything in hands, so let's try cryo-EM. And if we do it, we try to do it properly. So we took purified protein. You see it's highly homogeneous here on the size exclusion chromatography profile. It is highly pure. And this is the full length, right? This is 2CB also with this domain, which binds the phosphate and then gives it over to the glucose molecule one inside the cell. So we did nanodisk reconstitution. So we put it back into a lipid environment to have it as native as possible. And you see here, this is the MSP1E3D1 that we took. It has a size of about 12 to 13 nanometer. And both were pretty pure, right? We had a little bit of aggregation before, so we took the main peak and did cryo-EM. So this is the electron micrograph. So you see beautifully all the particles. And I have to say the 2D classes, they looked very crispy, right? If you look at the details, you see already how crispy this is, how secondary structure details come if you have a close look at this two-dimensional projections at these 2D classes. Okay, then this is Patrick. Patrick wrote, he took over this project as a master student and then pursued this as a PhD student. And he managed to get the structure out of this protein finally, right? So it was very intriguing because looking at the particles he saw that there are like two different populations, right? And as you see on the left-hand side, he could get a kind of asymmetric structure, right? As you see here, at a nice resolution of 2.9 angstrom. And from the same data set, from the same preparation, he could get a symmetric one at a little bit better resolution. You see 2.6 angstrom. So you clearly see that symmetric. That's a view from the membrane plane, right? And this is asymmetric. So this is now the electron density, of course, the Coulomb potential map. I'm a little bit stuck in X-ray crystallography, sorry. So you see the Coulomb potential map here, and then the model that was built, and we start really seeing with a symmetric structure that we have two domains, right? We have this dark blue domain and bright blue domain, and this is a transport domain, and this is a so-called scaffold domain, right? It assembles on the other side again with a second scaffold domain and again a transport domain. And this is symmetric, right? And it's inward facing. It's facing the cytoplasmic site. And the second one is more interesting because it's asymmetric, right? And this one has again the scaffold domain, everything is there. You have the transport domain. One looks the same as the previous one, but in this asymmetric one, the transport domain is different on the second protomer, right? And this one is outward facing. So we were very lucky and could get a structure of a particle, well, of this 2C transporter, which with one inward and one outward facing conformation. To our surprise, there was no B around, right? So we would expect that here there is some B domain, was very flexible. It seems to be an intrinsic feature of this B domain, just to be around, but not necessarily to make tight interactions with the transporter. On the right-hand side, you see the structure of this protein schematically, and it has 8 transmembrane helices, and really in the 90s, 2000, it was very hard to predict how many transmembrane helices this protein has. Many said 10, others said 11, others said it was a little bit an chaos. And no wonder, because if you have a look here, there are many split helices. There are like helical hairpins, several ones. There are several secondary structure elements that somehow messed up the prediction of the number of transmembrane helices, just because it is a quite complex architecture of this protein. Okay, so let's have a look at a little movie. I hope it works now. Before it worked. So again, you see the asymmetric dimer. You see in green and in blue, you see the 2 different protomers, right? Dark blue and dark green, you see the scaffold domains. They are pretty rigid, right? And then we notice that the transport domain is the part which is quite different, right? And it seems to be the part that somehow moves during the transport of the glucose molecule. Okay, so we had glucose bound, right? We supplemented the protein with glucose, and we were lucky. We had a beautiful density for the glucose. And here you see the model, right? We could identify all residues that interact with the glucose molecule in both in the inward and outward facing conformation. And basically, the interactions are the same, right? There's a little difference here and here, right? But basically, the domain just binds the glucose and more or less stays the same, right? We did also with Thomas Lemin at our institute some MD simulations. So you see here from the structure who interacts with which hydroxyl group or which oxygen atom. But here also you see, based on the intensity, who is making stronger, or you see here the hydrogen bond frequency. So you see, for example, that these 2 guys here, this glutamate and this arginine, they are interacting more intensively than this histidine with the substrate. So we correlated, although we also brought in a little bit of dynamics and not only this rigid view from the structure. So it is, as said, quite complex, right? Many residues interacting with the substrate, coming from different elements of the structure. Good. So here a little bit now to understand which residue is important, right? We know now which residue interacts with which hydroxyl group from the structure, right? So the next thing we could have done is to do an uptake assay, mutate all residues that we identified here, right? K to A, to alanine, glutamate to alanine, and so on. So this is really a marathon, really a lot of work, and this assay is really not so trivial because there is also this phosphorylation there, and to have this phosphorylation and the transport assay nicely running, you also need the other components to have overexpressed in bacteria. So it is not straightforward. It's not trivial, a huge amount of work. So we tried to be a little bit innovative here. So we took a protein, purified protein, and did thermostability using nanodifferential scanning fluorimetry. So you see here a curve, for example, in black. That's the control. That's the normal protein without glucose, without substrate. You go to the inflection point here, and then you get the inflection temperature. It's not a real melting temperature, right? Because you see here, you increase the temperature, you denature the protein, and what you look at is actually how this inflection point moves to higher temperature if it's stabilized by a small molecule like a substrate. That's very simple principle. So this is without D-glucose. If you add D-glucose, you get an amazing increase in the inflection point of 9.5 degrees, which is significant. It's a huge stabilization, right? And now what you can do is you can, instead of mutate the residues, we mutated, let's say, the hydroxyl groups. So we just used the oxyglucose at 1 position, 2 position, 3 position, 4 position, and so on, and not having the hydroxyl group, just look at it, right? And was very elegant. And what we see here is, first of all, the high stereospecificity. So L-glucose does not really stabilize the protein. It's like no ligand. Glucose 6-phosphate doesn't stabilize, right? Makes sense because it should not stay bound. It should leave the protein, so it doesn't like to bind. The affinity is low. Then if you take 1-desoxyglucose, right, this is like the mutation at that position, right? If you go here to this position here and you just remove this hydroxyl group and then you have a hydrogen, then you somehow mimic a mutation, but you don't risk that the protein is not expressed properly, or there are higher or lower amounts, so you have everything under control. And you see that there was also a very nice stabilization, right? So it binds, right, beautifully. Not as good as the D-glucose, right? But the 1-desoxy binds. So the protein doesn't matter too much what you have here, right? And this you get confirmed here using methyl glucose. So methyl also at the 1 position. So 1 position is quite flexible. You can add something at that position. However, the other positions are essential. You cannot remove that amino acid or that hydroxyl group. There won't be basically no stabilization, so it is really important for binding. We also tested a few disaccharides, as you see here. So maltose had a little bit of stabilization, and somehow it makes sense because maltose consists of 2 glucose molecules. But it was not so huge, but it was significant. It was there. So that was a nice essay just to see if the residues, right, really interact with the substrate. Okay, so looking at the structures, right, we have now this outwards facing protomer and this inwards facing protomer displayed here. We see here where the glucose is, right, here in orange. It is bound to the transport domain here, right? And we see that the whole domain somehow rotates down, right, towards the cytoplasmic side to release it. So this is more or less static, right, and just slides around the scaffold domain. So it has an elevator type transport mechanism. And what is interesting is that there is, if we compare the scaffold domains of the outwards facing and inwards facing, we found a position where there are these 2 prolines, which works a little bit like a hinge, right? And there is this hinge that makes then this, allows then this sliding down of the transport domain so that the substrate can be released into the cytoplasm. Let's have a look at this in a movie, right? And just remember here, right, you have this hinge that allows then this helix to go down, and somehow that the whole transport domain can move down and then expose the glucose to the cytoplasmic site where it is finally released. So that was very useful information from a mechanistic point of view. So we did also a lot of molecular dynamic simulations. I will not go too much into detail, but one thing I would like to mention is that looking at the structures, right, we just went down along the axis where the binding pocket is, right? And we noticed that actually our structures, right, if you look at here and here, they are in a occluded state. So to be correct, right, at the beginning we said, oh, it looks outward open, that's okay, right? But to be correct, it's outwards facing occluded, substrate bound state. Okay, so the substrate is trapped. It cannot easily go out because there is a kind of gate closing it, right? As you see here and here. So this is what we got actually from cryo-EM, right? We got an outwards facing and inwards facing occluded substrate bound states. Okay, and now doing molecular dynamic simulations together with Professor Thomas Lemin at our institute again. It was very interesting because he put that into a lipid bilayer and let the simulations run almost a cumulative time of about 5 microseconds. So that's not bad, right? Quite relatively long time. And what we observed is that at one point the glucose is leaving, and before it can leave, this opens up. So with the molecular dynamic simulations, actually using our cryo-EM structures and microsecond simulations, we could obtain models of the outward open, no substrate bound states, as you see here. So outward open apo and inward open apo states. Good. And just to summarize this first part, right, we got really very nice information from cryo-EM, but also from molecular dynamic simulations, and all these labels here in orange or dark orange here we could get from this study. These conformations we could not get, but somehow it makes sense. Glucose 6-phosphate, as you saw, doesn't want to bind. It just wants to be released, so the affinity is miserable, right? Makes sense. And according to the common view in the community, there might also be this kind of states which are kind of intermediate states between outwards facing and inwards facing. So outwards facing apo we got from the MD, also one of these states we could catch in MD. I did not show it to you. This is cryo-EM, again cryo-EM, and also the inwards open apo we could get from molecular dynamic simulations. So that was my first part regarding the transmembrane glucose transport in bacteria, specifically in E. coli, through this PTS glucose transport system. Now I would like to switch topic, right? As I said, I would like to talk a little bit more, give more an overview of what we are doing or what we were doing the last years, and also bring more examples instead of very detailed information. So the second part is about transmembrane light-driven proton pumping across the plasma membrane. Again, structural and mechanistic insights, and I would like to start with an introduction, and I'm always fascinated how long it took to get such structural information from this very, very interesting group and family of light-driven proton pumps. So everything started with these publications in 1971, right? And that's a very classic one. It's Oesterheld and Stokenius, right? And they described a rhodopsin-like protein, right? So opsins were known, right? The eye, vision. So they described a rhodopsin-like protein from a so-called purple membrane in a very bizarre organism, namely in Halobacterium halobium, or later it was renamed to Halobium salinarium. You might have heard one or the other. So these are halophilic bacteria, right? And you see them at different lakes, hypersaline lakes. You see here a beautiful example in hypersaline lake in Namibia, and you see this beautiful color, right? And this beautiful color comes also not only because of bacteria, right, which are very colorful. and this contain the so-called purple membranes, which contain this rhodopsin-like proteins which have an retinal bound. And then same issue here, right? There was a structure of the purple membrane. Well, at that time, you could write structure of a purple membrane. Today, you would expect a little bit more than a description saying that there was a molar ratio 1:1 between protein and retinal, and that the protein is about 26 kilodalton, right? So that was the beginning of a research which is still going on nowadays, right? Then here an example, in Basel, we were looking, of course, at this purple membrane, and in my opinion, there is not better than an atomic force microscope topography to show how such a purple membrane looks. And you see it consists of these trimers. You see here? Here you see one trimer, zoom out. So 1, 2, 3 protomers, 1 bacteriorhodopsin, 2, 3 bacteriorhodopsin. So there are 3 light-driven proton pumps which make up a trimer. And all this is a fantastic two-dimensional crystal which is highly ordered. And this is then called purple membrane because when you isolate it, of course, it has a beautiful purple membrane because of the retinal cofactor which is bound to the protein, and the whole environment, the whole change in electronics makes it then retinal is not colorful. Retinal is a little bit pale yellow, right? But if you put it into the right environment in a protein, it gets a beautiful color. And in this case, it's purple, right? So you see these two-dimensional crystals, right? And then Henderson and Unwin, you might remember Henderson, one of the 3 guys that got the Nobel Prize for cryo-EM a few years ago. So he made a lot of work with this beautiful two-dimensional crystals and electron microscopy. So actually, this here was the first view on a membrane protein, right? That showed that there are like alpha helical elements and not beta-barrel structure at many thought at that time. So really pioneering work. And only, now we are 71, 71, 75, and only then in 97, right, basically at the same time, there was the structure that came out by Kimura et al. From Yoshi Fujiyoshi's lab in Japan, who had the best microscopes, helium-cooled cryo-EM electron microscopes for electron crystallography. Finally, the structure came out of bacteriorhodopsin at a high resolution. There are some other publications. There are also lipid cubic phase work and some previous work by Grigoriev and Henderson. So finally, many years later, we got really a molecular view on this protein, and you see it consists of 7 transmembrane helices, and here you get this, you see the retinal, how it's bound. So this is the introduction, and that was all about an archaea, right? That made history, right? This archaeal protein, this bacteriorhodopsin. But only in 2000, it was shown that not only archaea use this light-driven proton pumps, right? But Oded Beja, he showed in this very important publication that there are also bacterial rhodopsin. So we had before the prototypical microbial rhodopsin from archaea, bacteriorhodopsin that I introduced before, and now discovered in 2000 only, right? They discovered that there are also bacterial rhodopsins, right? And that was the beginning of, with this second prototypical microbial rhodopsin brought from bacteria, right? So they live here in the sea, right? And depending on, they have different colors depending on the distance from the surface, depending on the light they have to capture. But the principle is just that they pump protons out, right? And this proton gradient is then used by the ATP synthase to produce ATP. Huh? So it's a kind of photosynthesis, right? But not the green one with chlorophyll base, but another one, very efficient one, used by microorganisms. Microbial rhodopsins, these are all bacterial. No, that's not all bacterial. There is also bacteriorhodopsin inside. So these microbial rhodopsins, they absorb light at different wavelengths. So it all depends on the environment around the retinal cofactor, right? On the change of the electronics in that environment that make this protein absorb differently. And this is very important because depending at which depth from the surface and bacterium lives, it needs to absorb shorter or longer wave photons. So very colorful, very nice to work with. Talking now about this bacterial, this prototypical bacterial microbial rhodopsin, which is the green light absorbing proteorhodopsin. I will abbreviate it now as GPR. So this is the prototypical one, right? So the first bacterial rhodopsin that was described, and I will describe, as I wrote here in brief, in very brief, the photocycle. So you see this is inside the pocket, right? You have here the cytosol, you have here the extracellular site, and we have here the cofactor, and it is in the all transretinal state. Then what happens, light comes in and then it makes a conformational change. It switches to the 13 cis retinal, and doing this, it gives, as you see here, I forgot to say that, this retinal cofactor is covalently linked to a lysine residue at helix 7, right, over a Schiff base. And this Schiff base is positively charged, and once it sees light at the right wavelengths and it goes from all trans to 13 cis retinal, it can then give over the proton to the so-called primary proton acceptor. I will come back to this specific residue in a minute. And then, very simply explained, there is also below a primary proton donor, in this case a glutamate here, which then recharges the Schiff base and it goes back into the resting state, which is the all transretinal. So that was very brief, just that you know the basics, namely that is coupled via Schiff base to lysine residue on helix 7. There is a primary proton acceptor, and primary proton donor I will talk less today. And like this, you have finally a proton pumping from the cytoplasmic to the extracellular site. So as you see here, since it's discovered in 2000, this only goes until 2018, there was an explosion in number of publications on this protein. And on the way to 2018, there was here in 11 a first structure of the monomeric green light absorbing proteorhodopsin done by NMR. And a second one from a kind of cousin from this green light absorbing proteorhodopsin, and the cousin is the blue light absorbing proteorhodopsin. This is just from an organism which lives a little bit further down from the surface, right, and absorbs rather blue instead of red light. And here, this cousin was found, right, and this is beautiful work by Hartmut Lücke and colleagues. They found by X-ray crystallography, they could crystallize it and get here and show that it forms a hexamer and a pentamer. And that was the knowledge at that time, but basically no high resolution of the green light absorbing proteorhodopsin. This was a nice start, but comparing later with available structures, it was not so waterproof, but a good start. So there was also large discussions on the oligomeric assembly of this protein, right? So you see here, this was solved as a monomer, and here as a hexamer, as a pentamer, the cousin of the green light absorbing proteorhodopsin, and the literature is full of publications saying it is a pentamer, and the others say no, it's a hexamer, and others say no, it's both. It can be both. So, and as you see here, using numerous different methods, techniques, there was no clear view on the oligomeric state of proteorhodopsin. So, pentamer or hexamer, that was also one of the questions. So we started pretty much to 2018 around, something like that, with the green light absorbing proteorhodopsin, and this is Stefan Hirschi. You will see a lot of now beautiful work by Stefan Hirschi. So when you express and purify the green light-absorbing proteorhodopsin, the wild type in E. coli, expressed in E. coli, and you purify it by affinity chromatography, then you see this. You see that there are like two bands. There is not only one band, right? So you see an abundant band and less abundant band. There are two forms there. And that's why we said, okay, this is a form which still has a so-called signal peptide attached, right? Which is cut off during maturation in E. coli. So that's why we have two populations, one with a signal peptide and one without. So we say, okay, let's make life a little bit easier to start understanding, or perhaps or not, this system, and we just cut away this signal peptide. And if we cut it away, we get very homogeneous protein only with one band, only one population. So again, we did not introduce a tag to avoid any potential histact induced oligomerization, just to be sure, because in the literature it's so controversial, right? All are histact proteins, or basically all, and they observe different oligomeric states. So we said, okay, let's cut the signal peptide and let's not introduce an affinity tag, just to avoid potential problems, right? And then we purified everything by conventional chromatography, like Ewing ion exchange chromatography. Luckily, the protein is very colorful, right? And we can detect it easily. So we could purify it by ion exchange chromatography. And then doing size exclusion chromatography, we could show that it's a beautiful oligomer, right, eluting at low elution volume, and there was also a population eluting at higher elution volume. And you see here the size exclusion chromatography column, and you see here beautifully how you really see by eye the separation of the monomer and the oligomer. So we have an oligomer here, and here we have a monomer, right? At that time, no cryo-EM, well, not on our side at least. So we were still trying to grow 3D crystals. We could not get any crystals from the monomer, right? However, from the oligomer, we could get beautiful crystals, and you see also this beautiful color coming from the cofactor. So that's one of the most beautiful crystals we could get. It was around 100 microns, a pretty large one. Stefan did a lot of work, screened a lot of conditions. We went a lot of time to the synchrotron, right? But that's what we could get out, right? Not more, not less. And as you see here, so the diffraction is pretty modest. You see diffraction spot that's 4.8, and it's anisotropic. So that was a little bit the best what we could get out, and very disappointing, of course, for us after so much work. So overall, we could process the data set. Overall, we had about 5 angstroms resolution. We could use one of this blue light absorbing proteorhodopsin structures for molecular replacement, and we could get something out of it. And you see here, ours are pentamers, as you see here on the top view. Here are tilted, so we could see the transmembrane helices and that it is a pentamer, but that's it, right? Data set was low resolution and anisotropic. So why was it anisotropic? Because if we look at the here you see the symmetry of the protein and the space group. What you see here is that in one direction it's beautifully packed here, making interactions, but here you have the detergent belt, right, of these two columns, and somehow they slide a little bit. So that's where the anisotropy comes from, and this just limited us, and we could not go to high resolution. However, Stefan did not give up, right? And we decided to express it now with a his tag, and he could get it pure, as you see. Again, it elutes at 11 point something. using this SuperDex column. It's very similar to the untagged one. And now, finally, we could get the 3D structure from this prototypical bacterial proteorhodopsin, and you could solve it at 2.9 angstrom. You see here the Coulomb potential map from the side, from the top, and it's a beautiful pentamer, right? Also, the helices are very nicely resolved. You can see here the phenylalanine, for example, and also the retinal was beautifully resolved. Please have a look at this methyl group. So here of the retinal, you could really discern them. Good. So finally, the structure of the green light absorbing proteorhodopsin, the prototypical one from bacteria. This truncated version without N-terminus, right, forms beautiful pentamers. You see here from the side, this is looking from the membrane plane. You see the retinal. You see again here a zoom in. You see here that there are hydrophobic residues. I will show you also later how the whole binding pocket looks with hydrophobic residues. And here is the proton translocation pathway. And here we have the D98. This is the primary proton acceptor. I explained before we have also nearby a second aspartate, and both somehow are a little bit the counter ions to the positive charge of the Schiff base by the lysine and the retinal. And what we see also is a beautiful geometry, right, given by these two tyrosine molecules, which beautifully orient this counter ion here, this aspartate in 3D, so that it's perfectly oriented as it should be near the Schiff base, retinal Schiff base region. A little bit a closer look and comparison with bacteriorhodopsin. You see again what I just said before. You have here the lysine on helix 7. It is Schiff base here. You get this. You have these two tyrosines I said that beautifully orient this counter ion. There is also some help by this arginine here in charge stabilization. But what is interesting and special is that the primary proton acceptor here is in close distance to a histidine. And this histidine was shown to influence the pKa value of the primary proton acceptor. You can determine that easily because it changed the color, right? You can titrate and determine this primary proton acceptor. And this interaction with this histidine, which is absent in archaea, as you see here, right, increases significantly the pKa of the primary proton acceptor, right? And you see here the pKa here of the green light absorbing proteorhodopsin is around 7.5, so at physiological pH more or less, because of interacting here. And once you remove this here, right, it is much more acidic, right? In the meantime, we have also the structure and data on an intermediate state, but it's not published yet. So this is just to have a look at the molecular level at this retinal Schiff base and how this region then can tune the pKa value of the primary proton acceptor, which is also at the end very important depending on the environment of the bacterium, right? If it lives in an acidic or in a basic or in a neutral pH environment. So I'm coming now to the last part, right? I'm over time, I have the impression. Luckily, I deleted some slides this morning. So the last topic, right, is about oligomerization. And there is this very nice study by Shibata. And there you see summarized that there are numerous microbial light-driven proton pumps which form trimers, including bacteriorhodopsin from archaea, and you have then others, right, from bacteria mostly, that form pentamers. And we also had a pentamer for the green light absorbing proteorhodopsin. However, there are also a lot of studies saying that it's a hexamer also. It can also be a hexamer, right? And there's a big dispute. The guys saying, no, it's a pentamer. The others saying, no, it's a hexamer, right? And others that say, no, it's both. So let's see what we found out, right? And what we did is we introduced this point mutation, which was described in the literature. And this point mutation at the N-terminus makes that the signal peptide sequence is not cleaved. That means we don't have these two populations of the wild type expressed in E. coli that I showed you before, one which was cleaved and one minor, it was about 30% population, which has still the signal peptide sequence, right? With this, you have only full lengths. Good. And how does it look? If you look at the structure, and again, Stefan, very happy to have done this, right? We published that last year. What he saw was something interesting. So when you have the full length protein, right? I just go quickly back to remember you. Right here you have the wild type. So now what you have is only this upper band here. You don't have any cleaved anymore, right? That's all about this mutation, right? So if you have this, then you have a mixed population in your purified protein. And you have pentamers, right? And hexamers. And he solved the structure from the same set of images and same purification. He could solve at 2.8 angstroms the structure of the pentamer and of a hexamer, right? Here he could impose a 5-fold symmetry axis, but careful, here it's not a 6-fold symmetry axis anymore, but a 2-fold symmetry axis. Because what we saw is that the N termini somehow embrace of 2 protomers and then making this kind of dimer, and then monomers can join. But we didn't see any N termini from these 4 other protomers, only from these 2. So we speculate that once it's synthesized in the membrane, these 2 can find each other and then the others can join, and then you have this hexamer, which has a 2-fold symmetry axis. So because of this N-terminal helices here. So that was very interesting, just that you have an estimate about 70% formed pentamers in our system and about 30% of the picked particles were hexamers. So very special interactions, right? Numerous interactions. So you see protomer 1 with protomer 4, as I said, somehow this N-termini embrace these helices, right? Forming this dimer. And then you have also the other 2 protomers. I only show the upper 2, not the lower 2. It's the same. So you have interactions with all protomers with the N-terminal signal peptide from protomer 1. So protomer 1 here in blue, you see the N-terminal signal sequence. It interacts with protomer 2. It interacts specifically with protomer 3, and of course with protomer 4 through its N-terminal signal peptide. And as you see here, very, very, very specifically. Okay, so there was one review, a very smart person. He asked, yeah, but that's beautiful, right? But what's the difference between the hexamer and pentamers? Good question, very good question. Very, very, very difficult question to address. But we did our best, and I have to give credits to Noor Ayub, which in my opinion did really a beautiful work during this revision, because you cannot separate by size exclusion chromatography a pentamer and a hexamer. Just see the mass is about, it's a small protein, a little bit more than 20 kilodalton. So how do you want to separate a pentamer from a hexamer? So that was impossible, right? A very difficult mission that was somehow solved by blue native gel electrophoresis. So there we could resolve the pentamer and the hexamer, right? Using this gradient gels, blue native gel blue native page, and you see here's a control, the truncated version, making only pentamers, and here the A218L mutant, which only makes full length protomers, right? But you clearly can see that you can separate pentamers from hexamers. So what Noor did is then he did that large scale, let's say. He isolated the bands, he removed the Kumasi and recorded on one hand side adsorption spectra. On the other side, he checked by nanodifferential scanning fluorometry the stability of this oligomers. And what he saw is that basically absorption maximum did not change. So absorption maximum was still at 525 nanometers. That is the absorption maximum of the retinal bound to the protein, right? And no difference between pentamers and hexamers. However, he could see a difference of about 5 degrees in stability. So the hexamer was less stable than the pentamer. Okay, that's all we could learn. We could not learn more about a potential difference, functional difference between pentamers and hexamers. That's the current state of this. So that was not enough, right? So having this in hand, we just wondered, okay, what happens if you express the green light-absorbing protein without a retinal in E. coli, right? Does this protein express, or is this degraded? Does it always need the cofactor or not? So this was done. It was expressed in is what is expressed in E. coli without retinal, and that's important because we know that in nature there are numerous organisms which express such a microbial rhodopsin, but has not the synthesis pathway to synthesize retinal, right? So where does the retinal come from, right? And why do they have it if they don't have produce retinal, the organism? So that was very intriguing, and also intriguing was, is it stable, this apostate, right? And that was again Stefan, and Stefan finally also solved the structure of the pentamer, right? This is the truncated version that only yields pentamers at the 3 angstrom of the apostate. That means this has no color, right? Could be purified by affinity chromatography like a normal protein, and he could solve the structure and could show it is a pentamer. However, it was very interesting because he saw a density there. But it's not retinal, it's not colorful, it does not look like retinal, the density, right? So that was very intriguing finding, and thanks to Professor Jürg Gertsch and Daniele Pellegata at our institute, they found that by mass spectrometry, also quite sophisticated one, that this molecule was decanoate. So this molecule, this density here originates from decanoate, and we build it in here and makes also a beautiful salt bridge with the lysine, which normally forms the Schiff base with the retinal. So that was very intriguing, and here you see all this beautiful hydrophobic pocket that also can bind the retinal. But in this situation, it binds this fatty acid, right? This decanoate. Again, with Thomas Lemin, different molecular dynamics simulations, considering the experimentally obtained density, and this just gives a high correlation in function of the density. So everything which is on the right is most probably fitting into that density, and you see also here the decanoate nicely fitted. But also you can see that others, depending on the organism, another molecule might take this place, this placeholder, in the absence of retinal. So what we think is actually that this, well, I will come to that to the end. Let's first have a look at the dynamics. So if we look at the structure of this proteopsin, that means without the ligand, without the cofactor, if we take this structure, we take also the structure of proteorhodopsin, but we remove the retinal, and we do this MD simulations. What came out is, looking at the simulations, this helices here, transmembrane helices here at this corner, are very dynamic, right? So they move a lot. I will not go into details, but just as a take-home message is that this is moving quite a bit in the presence of decanoate, right? If you remove decanoate, and if you remove retinal, this here basically collapses so that nothing can go in or out into that binding pocket of retinal. And if we have retinal bound, we have a situation which is a little bit in between. It's not collapsed, but it's not so open as decanoate. So what we think is that decanoate somehow stays there in the membrane of this bacteria, waiting that from the environment, right, a retinal comes one day and then partitions in the lipid bilayer and then finally binds to it, right? And this is very probable since retinal is a derivative of vitamin A. So it is found, but these guys, we think, produces protein in the membrane, and they wait until this retinal comes from the bulk outside in the sea. So looking at the... Yeah,
many more slides?
No, that's the last one, I think, right?
Okay,
yeah,
perfect.
Two more, sorry.
Sure.
So just to finish, right? This is our structure of rhodopsin. And here, what we saw is that there is a kind of entrance between helix E and F. So there is a kind of opening, right? Where decanoate could go out and retinal could go in. This is interestingly also observed in this GPCR here, which is opsin. But this one has not only this entry here that we also observed, but it also has a second channel, right? And this makes sense because opsin and rhodopsin, just every time it sees a photon, it just does not make only an isomerization, the retinal, but it also leaves the protein and is regenerated and then comes back. Quite complex process. And there is also a channel rhodopsin, which also was solved in the apostate and also looks as it has a similar channel as here, right? So what we think, to make it short, is that here this is the entry point for decanoate and again exit point, and also the entry point for retinal to finally have the functional proteorhodopsin. So this is my last one, right? This is a summary. So what we think is that we have folding, insertion, we still have the signal sequence here, right? In early stages, a placeholder might come. This protein can easily be acquired from the fatty acid synthesis. Then it is cleaved off. It makes oligomers, either pentamers or hexamers. What the exact function of this is, we don't know yet, right? And then at some point, for those that do not synthesize retinal, it comes from the outside, from the bulk, partitions and binds to it, and then it's functional. And that's it. Sorry for having prolonged. I didn't expect to talk so much, especially early in the morning. And just this final slide to give credits to all the people. I mentioned MD by Thomas Levin, mass spectrometry by Jürg Gertsch. These people always contributed to all projects I showed. And big credits to Stefan Hirschi, and also big credits to Patrick Roth for these two presented projects. Thank you very much.
Thank you very much, Dimitrios. So we might see if anyone has any questions. So if you can either put up your hand, and you can either or in the chat. So there's two questions already in the chat from Patrick. So the first one was, did the MD simulation also aim to simulate the flexible domain of the glucose transporter? So that must be domain B or 2B. And then also, what were the concentrations in the nano DSF screen of the glucose analogs?
Yes, glucose analogs, as far as I remember, was 10 or 20 millimolar. So it was quite high, just to have it really, really saturated, not to miss something. And about the MD, well, MD, we would need much more time to reach a point where perhaps it goes from one conformation to the other, right? From outwards to inwards, for example. But this almost cumulated 5 microseconds only could give information, let's say, on the release of the glucose molecule. I didn't talk about that, but there were also experiments trying to bring back the glucose molecule into the binding pocket just by positioning it nearby and doing the simulation to look if there is a binding event. And there was a binding event, not often, but there was one. And yeah, that's more or less what we could get out of it, right? On one hand side, the hydrogen bonding frequency that I showed, that means the amino acid residues interacting with the glucose, this opening of the occluded state into the outward open, so gaining two more structures, let's say, or conformations. I think that's more or less what we could get out, and I think it's okay, right? Perhaps in a few years we could get the whole cycle simulated, but for the moment we are far away.
Nice. Okay. There's another question in the chat from Sol. So it was about, were you able to get cryo-EM structures / populations of the 2C transporter without glucose bound? If not, why do you think you can find the non-glucose bound structures in MD but not cryo?
So can you repeat the first part? So why there was
Yeah.
no, if we tried without glucose, right?
Yeah, well, could you get structures without glucose bound? So perhaps in your same data set where you've
Yes.
supplemented from glucose, was there any without glucose?
No. So if we added glucose, and I think it was around 10 or 20 millimolar, only glucose bound, right? Of course, we tried also without glucose, right? And without glucose, it's not published yet. We only got inward facing. So the dimer both are inward facing. And what we published this year was a detergent solubilized structure. These were all in nanodiscs, right? Just not to have detergent. And this structure that we solved in dodecyl maltoside, so a very common detergent maltoside, was very interesting. I didn't want to show it. As you see, I don't have any time anymore anyway. But what we saw is that the maltose from the dodecyl maltoside bound at the substrate binding site, and that the alkyl chain of the dodecyl maltoside somehow trapped the 2C transporter in an intermediate state. So also very interesting, but again, I could not talk about everything today.
Okay. Sounds great. Did anyone else have any questions that they wanted to put in the chat or put their hand up? I don't see any at this point in time, so maybe in the interest of time, we'll just leave it there. And I would like to really thank Dimitrios again for his lovely talk on very different membrane proteins. And just a reminder to everyone else that our next CCE MMP seminar will be on September 9, where I'll be giving a talk actually. So looking forward to seeing everyone then. So thank you again, Dimitrios. It was lovely to hear your work, and thanks everyone for coming.
Thank you very much. Bye-bye. Thank you.
Bye.
Thanks, Dimitrios.
Thank you.
That was a whirlwind tour of a lot of work. That was
Yeah.
really interesting. Thank you.
So have a nice day. Bye-bye.
You too.
You too.
Bye.
Bye. Thanks.